multiple sequence alignment. So, multiple sequences alignment is very important for constructing phylogenetic tree, all these things. So, multiple sequence alignment is a very important parameter for constructing phylogenetic tree. All. So, we are moving into multiple sequence alignment. One of the major tool which we use for multiple sequence alignment is cluster omega. So, I'll just show that cluster omega is page is like this. So, what we can do is that we can just paste our sequences. I have just taken it for the phylogenetic tree construction only. I have just taken it from NCB and just pasted here, and then we can give that same. Actually, multiple sequence alignment, that algorithm can take only up to 20 sequences at a time. For 20 sequence can only be multiple sequence alignment means comparing more than 2 sequences at a time. So, that is a multiple sequence alignment. We can use up to 20 sequences. So, algorithm is has limitation up to 20 sequences. So, only 20 sequences can be compared by the cluster W or any other multiple sequence alignment tool which is available now. So, what it is doing is that actually the multiple sequence alignment, actually we can do it with a dynamic programming algorithm such as Needleman-Wunsch algorithm. How they do is that we are in Needleman-Wunsch algorithm, we can see a 2-dimensional array. So, in multiple sequence alignment, depending on how many number of sequences are there, we will create an n-dimensional array. N-dimensional graph we will create, and we will build up the alignment. That is what they are doing in the multiple sequence alignment using dynamic programming algorithm. Usually it is a 2-dimensional x and y axis, and in multiple sequence alignment using dynamic programming algorithm, they will construct a n-dimensional array, and we will compare them. So, that n-dimensional array means it is computationally it is too much very tedious task. So, we can only compare 20 dimensional array. We cannot go greater than 20 sequences in the multiple sequence alignment. But cluster W work in a different way. I will tell that. When we go with a dynamic programming algorithm for multiple sequence alignment, comparing more than 2 sequences, we use a n-dimensional array. We usually use a 2-dimensional array in that Needleman-Wunsch and Smith-Waterman algorithm which we have seen today, and in the case of multiple sequence alignment, we use a n-dimensional array in comparing the sequences. But in the case of cluster, the algorithm is bit different. I will explain that. So, we are just put our sequences here, and we will just submit our sequences, and they will filter, and we will their job is the job is queued, so they will give the result. And cluster W or cluster omega, both are of same only, and they have renamed as cluster omega. Earlier it was cluster W. The algorithm was first described by D.G. Higgins and P.M. Sharp in 1988, and they begins with aligning closely related sequence. Usually what they do is that they will find out the closely related, they will construct a guide tree, they will construct. Guide tree is that in the guide tree they will find out the closely related sequence, GUID. Okay, anyway, the guide tree they will construct, and this will tell the closely related sequence, and then they will align. What they do is that they will align the closely related sequences first, and then adds the increasingly divergent sequences. So, first they align the closely related 90% similarity sequence, 80% similarity sequence, 70%. So, they will add the less similar sequences next to that alignment. So, they first align the closely related sequence, and the closely related sequences were found out using they will construct first a guide tree. Using that guide tree, they will find out the closely related sequence, and closely related sequence are aligned first, and less closely related sequence, divergent sequence were aligned later. So that is what it is doing. The cluster W is doing. So in the cluster W, they will construct a phylogenetic tree first. That is a gate tree. They can say as a gate tree, and they will determine the similarity between the sequence. And using that guide tree, using that tree as a guide, you can see this is called as a guide tree in the cluster W. The closely related sequences are aligned two at a time using dynamic programming algorithms by pairwise alignment. So closely related sequences are compared two at a time, and they will build up their similarities. So closely related sequences are aligned two at a time using dynamic programming algorithm. So then the less closely related sequences were aligned later. So they are usually these alignments were scored by PAM and Blossom matrices, and based on the score only the particular result is coming. So like that the alignment is done. So they will build up a gate tree, and they align the closely related sequence two at a time by pairwise alignment using dynamic programming algorithms. That is what and they will show the result of the cluster W result. You can see the cluster W result. You can see the alignment between the sequence. Here we can see the gaps in the sequence, and here the alignment, because the different all the sequences are of different length, so they have aligned all the sequence which we have given, and you can see in the alignment several symbols. You can see some dots, some colon, some stars, all these things. Usually it denotes the alignment, the quality of the alignment. What these symbols indicate, we will just see. The star symbol indicates a perfect alignment. The star symbol here indicates a perfect alignment. The star symbol indicates a perfect alignment. Okay, here star is there. Star symbol indicates a perfect alignment. And then this colon, this symbol indicates the site belonging to group exhibiting strong similarity. This symbol indicates the similar amino acids are present here. Similar amino acids are present. Similarity can be seen. And then this full stop dot symbol indicates the weak similarity. We can say there are this dot symbol are there, are showing the weak similarity. So different symbols we are seen in the alignment which say about the extent of similarity between the sequence. So cluster W, and usually they will construct a guide tree before the construction of the alignment, and then they will construct a phylogenetic tree also, and you can see the phylogenetic tree. So you can see how they are aligned, and tree data, all this. We are going to construct a phylogenetic tree with the same sequence, and they are using neighbor joining method for the construction of phylogenetic tree, and we are going to construct with another tool called Mega. So this is about cluster W, and we are going to construct the phylogenetic tree. So cluster omega uses seeded guided trees and hidden Markov models, HMM profile techniques to generate the alignment with two or more sequences, three or more sequences, or two or more sequences we can say. Biologically meaningful multiple sequence alignment, it produced biologically meaningful multiple sequence alignment for divergent sequences, and evolutionary relationship is seen via they construct cladograms or phylograms. This tree they will construct is what is the difference between phylogram and cladogram? Usually the phylogram means it is a tree. You can see here phylogram, the branch length will be different. The branch length determines the evolutionary closeness between the two. So C and D are branch length is very less, So they are closely related than A and B. So the branch length, but A and B, this cluster is very farther related to the C and D. So the branch length in the phylogram determines the closeness between the two individual or sequences. In cladogram, the branch length remains the constant. That is the difference. So phylogram, the branching is a branching diagram or tree assumed to be estimate for phylogenetic study. The branch length are proportional to the amount of inferred evolutionary change. So how much evolutionary change is there, depending on that, the branch length will change. But in the cladogram, assumed to estimate the phylogeny, where the branch length remains same. Branch length remains same for cladogram, while branch length remains different depending on the evolutionary relatedness. The branch length will be different for a phylogram. So we will just go through phylogenetic tree. We can see it's a phylogenetic tree. The various parts of phylogenetic tree. When we are going to phylogenetic tree, the various parts of phylogenetic tree, there will be a root will be there. Usually we will, when we are constructing phylogenetic tree, we will go for an out group, the sequence which is negative control. In experiment we will take negative control. For the construction of phylogenetic tree, we will take a sequence which is unrelated to the others. So usually in phylogenetic tree construction, we will take a sequence which is unrelated to the other sequence. So as out group or a negative control, and these are the in group. These are the sequence which we are already going to compare, and there will be branches are there, and roots are there, and these are nodes are there. So branches are there, and nodes are there, and then roots are also there. So this build up the phylogenetic tree. So we will just go through the construction of phylogenetic tree. So we have this file. We have taken the, I think 10 sequences or 7-8 sequences are there. Same only exokinase I have taken. I have just taken for showing you people. So we will construct a phylogenetic tree. First we need to open a tool called BioEdit. So I will just take BioEdit. I will just search and take BioEdit here. BioEdit. So I will take BioEdit here. Let it open. BioEdit is a tool. I am not going to use this tool for phylogenetic tree construction or anything. I am using this tool for cluster W alignment. Usually the alignment which we get from the cluster W tool is not being accepted by the I have tried sometimes, but it is not accepting properly by the Mega software. So I used BioEdit for alignment. I think alignment you can do it in Mega also, and Mega it is Mega the phylogenetic tree all we can't download. We need a paid version. We can I will just open our sequence. So this is the BioEdit. This is how the BioEdit look like. So it is used for the editing of DNA sequence, RNA sequence, protein sequence, etc. So we can edit the sequences here in this tool, and we can just open our file by clicking open, and that file is at desktop. This is the MSA file. I can open here. You can save it in a notepad or a word pad. You can see the sequences here, and different colors is given because of the different properties of different amino acids. Different colors is being represented here, and you can go slide the sequence and see the whole sequence, and we can select the sequence, and we can we can slide and see the whole sequences, and I'll just see whether the whole sequence are aligned properly. I think I have taken only very similar sequences. Let me check. So some of the sequences are lengthy, so if you want to make the sequences of similar size, you can select the sequences and delete that. You can just go here, the mode, and just you can click the edit, so that you can just select the region, selective region. You can delete it. Bigger figures, this much portion you can select and just delete in your delete button in your system. Just click that delete, so that that sequence will be deleted, so that the sequence can be made in a uniform way. If you need, depending on your how depends on your work, what work you are doing, you can edit. I am not just telling about phylogenetic and alignment, it's just some second only to take. I am just saying you don't need to do this. Depending on what work you need, you can just edit the sequences using this, and you can insert gaps. If you are working on some other work, you can insert gaps in the sequences. You can click this eye and go to any sequences, and you can you can insert gaps in the sequence. It is not working. Sorry. Yes, right clicking only, insert gaps where we can click here, right clicking only, the gaps will be inserted. You can see here. So the by right clicking, the gaps can be inserted here. So gaps can be inserted, and gap look like a wavy nature. You can lock the gap, so that the gap can convert it into lines. You can see the lines. So you can unlock it, so that it becomes that wavy line. So depending on your work, you can insert the gaps and delete the gaps by the right clicking. You can delete the gaps, and as well as if you are studying mutations, etc., you can add we can do shade the identities and similarities in the alignment window. You can find out the identities in the sequence. So you can see some region of the sequence only is identical, that highlighted region only the identical is identical, and we can see the identities and similarities between the sequence, between the whole sequence. That region only is seen identical. So if you are studying mutations in DNA, all these things you can find out. So different tools you can utilize, show features of the sequence, that is already shown, because different colors, they have indicated different properties of the amino acids. And then so we can then what we can do, normal view mode, black colored view mode, information based on column shading. So column shading can be done, and we can click here, and we can go for normal mode. Someone is there, so like that we can do. And then where is BioEdit? So these things, lot of edits can be done. Can go for monochrome color, different color, and we can go for drag and grab and drag the sequences using this tool, mode grab and drag. We can grab the sequence like this, grab the sequences in blocks. So we can grab the sequences, and like that we can do, and we can edit it. We will insert gaps here. So we can grab the sequence for different sequence editing. I am not saying about a phylogenetic tree or anything, just for sequence editing. And for nucleotide sequence you can try the similarity that button, so that you can find wherever the changes occurred, and you can, when you are studying mutation etc., it will be helpful for you. We have taken the amino acid, so there is no much changes have been seen or similarities seen in some regions only. So like this, lot of things can be done, and there are accessory application. You can do blast with this, and you can do cluster W. I am just doing cluster W. You can construct the tree also with these different methods, but we are constructing using Mega only. So we are just clicking the cluster W multiple sequence alignment. So actually in multiple sequence alignment, when we are doing the first part of the construction of the tree, so they are asking for the neighbor joining tree bootstrap. They are constructing a tree, and then only they are finding out the multiple sequence alignment, as we have seen. So bootstrapping need to be done. How many number of bootstrapping need to be done during the construction we need to give. Then we need to study what is bootstrapping. So bootstrapping means usually during the construction of phylogenetic tree, multiple sequence alignment itself, they will construct a phylogenetic tree. During the construction of phylogenetic tree, they will construct a phylogenetic tree multiple times. As we see in the sequencing, we will sequence the particular piece of DNA thousands of times in order to get a confidence, in the sequence. So like that, they will construct the phylogenetic tree multiple times. If they construct the phylogenetic tree 1000 times, and then they will we can say that the bootstrapping value is 1000. How many times the tree is prepared and compared each other, the confidence of the tree will increase. So if we go for a higher bootstrapping value, we will get a perfect tree. If we go for 100 bootstrap, means it is the tree will be prepared 100 times only, so errors in the tree can occur. So we asked many times, in the experiment, you people are doing duplicates, triplicates, more than that. If we do triplicates, we will get the correct result. So like that, if we do 1000 times the building of a tree, 1000 bootstrapping, we will get a perfect tree than a 100 bootstrapping. So the tree will be constructed 1000 or times or more. That is called bootstrapping. So we are just giving the bootstrapping 1000 here. It's a default one, and we are just going to run the cluster W here. So we are just running the cluster W. Our time is just going, but we will complete. So we are just running the cluster W, and we got the alignment file here. This is the alignment file, and we will just save it. Save as. So we will just save in the desktop only. So alignment, I am just winding up just for construction of phylogenetic tree only, and desktop we are just saving it in the FASTA format, and then we will go to Mega for the construction of phylogenetic tree. Just some second only. Mega, the tool is not fully free, but we can construct a phylogenetic tree. We used to take a screenshot or a print screen, and we used to take the picture. Otherwise, saving is very difficult. We can open our file, our file here, which we have saved in desktop alignment, or like that we have saved it. It is not there. Yes, BioEdit file, exokinase is there, not that. So alignment file, this is the one. No, I'll save once again. Let us save. Let us save once again. Save us. I will save us alignment. This is alignment, that only I can save us first only. Yes, I think it will come. hope it will come. Let's open the alignment file, else we can save it in some other yes. So we don't need to analyze it, we just align that. So we can see the alignment of that sequences here, they will show. So the alignment of the full sequences there, the mega software will show. And our main thing is for the construction of phylogenetic tree. We will go to phylogeny and construct different types of trees are there. There are different types of trees are there. Usually the trees can be character based trees are there, and maximum parsimony, maximum likelihood trees are there. Usually for when we are comparing very high number of complicated sequences, we go for the neighbor joining tree, and another tree which is similar to the neighbor joining is the UPGMA tree. So what it is, the tree, we will see it. So there are different methods for constructing the tree. The distant based trees, distant based methods for construction of trees. So the distant based methods include the neighbor joining as well as the UPGMA trees are constructed by distant matrix method, and then other trees are there, maximum parsimony trees are there, and minimum distance trees are there. So these are some of the methods which we use for constructing a phylogenetic tree. If we have the multiple sequence alignment, by using these methods we can construct the phylogenetic tree. Distant matrix method, such as the methods such as Fitch, Margolis, neighbor joining, UPGMA, these are the methods which uses distant matrix method for construction of phylogenetic tree. Distant matrix is calculated from the sequence data set. It's very simple, and this distant matrix method find only one tree. Somewhat old fashioned, this if our alignment is good, evolutionary distances are short, we use for distant matrix trees, mostly used for constructing large data sets. If you have a large data sets, we use for neighbor joining or UPGMA method. It's old method, it constructs only one tree based on the distant matrix they construct. And when it comes to maximum parsimony, the maximum parsimony, they will construct a multiple tree and finds the optimum trees by minimizing the number of evolutionary changes. So they will not, the maximum parsimony method will not take into account of evolutionary relationship. Evolutionary pattern will be minimized, or evolutionary relationship between sequence will be minimized. No assumptions on evolutionary pattern will be taken in this maximum parsimony. So because they are not considering evolutionary relationship between the sequence, they will oversimplify the tree, and they will produce several good quality trees, and they find out the optimum tree. But as they are not considering the evolutionary relationship, is one of the drawbacks of that. The maximum likelihood, not the minimum likelihood, I think, sorry, I said it's minimum distance. So this is maximum likelihood. So the maximum likelihood tree is that this is the best tree. They will go for multiple trees, and they will find out the best trees with including the assumptions of the evolution relationship. So nucleotide models are more advanced at the moment than amino acid models. So usually this maximum likelihood is used for nucleotides, construction of nucleotides, than for proteins. And the program requires a lot of capacity in the system to execute this maximum likelihood programs. So these are the methods. Mainly we use distant matrix method, which is one of the oldest method. There in this method we will construct only one tree, and these used for constructing large data sets. Maximum parsimony will not consider evolution relationship between the sequence. So they will oversimplify evolution, and is one of the drawback, and they will construct multiple trees and they find out an optimum tree. The same maximum likelihood also construct multiple trees and find out a best tree, but they are need a lot of system capacity to run this maximum likelihood, and they are now mostly used in comparing the nucleotide sequence rather than the protein sequence. So this is all about our sequence alignment. So we will construct the tree using mega now. So we have taken phylogeny. We will construct using a neighbor joining tree. We will construct. We will click the neighbor joining. So we need to give. We will use this sequence only. So what is happening? We will construct a neighbor joining tree. They are asking to. I think we need to have a BioEdit file. I think we were somewhere wrong. I think we have a exokinase file there. I will just pull that. The mega got crashed. Anyway, something happened to the mega. I will take it once more. Let it off. So this is all about the workshop. We will do it mega. So if you have any feedbacks about the workshop, you can tell it now. We will do the mega now as it is getting load. Any feedback or anything about the workshop, have you learned something? You can tell if you are interested to tell, or you can please write down the feedback in that. I think we can construct a phylogeny tree. For that we need a mega file. We can take a FOSTA file here. This file we can take. Hope we can construct. Yes, we are taking the protein sequence only. We can open the file which we need for the phylogeny tree construction. We can, it is a protein sequence only. If it is nucleated, we can sell that, and these are the various parameters for the construction of phylogeny tree, and these are the tools. These are the various parameters. You can look on that. Test of phylogeny and phylogeny tree construction. We are using the statistical method, neighbor joining method, and several parameters used for the construction. You can just click the compute. The tree will be generated very fast. They will generate the tree. You can get the tree. You can see one of the outgroup here. That's again is from a bacteria I have taken as outgroup, and these four are Drosophila. They are closely related. They can be seen in a one branch, and you can see the other hexokinase of other organisms. And these two hexokinase from Rhizoctona and this Diplocapod, they are closely related. They are seen in other branch, and you can see the hexokinase from you can see the evolutionary distance between hexokinase from different organisms. Same hexokinase only I have taken. Hexokinase from bacteria, Taponema. This bacteria is seen as a outgroup or negative control. So you can see the details of the tree. You can send these details when you are publishing the data, and you can show these references when you are publishing the data in your research paper. So like this only we constructed the phylogeny tree.